to have the protection.
Okay, now.
Now. So it's my great pleasure to introduce our dear friend, Seamus Martin. Seamus holds the Smurfit Chair of Medical Genetics within the Smurfit Institute of Genetics at Trinity College Dublin, Ireland. And he has an international reputation in the field of programmed cell death and is one of the most highly cited scientists in the world on this topic. Seamus obtained his PhD at the National University of Ireland and has held postdoctoral fellowships at University College London, UK, and the University of California, San Diego, USA. Seamus' achievements are numerous. He was elected to the Royal Irish Academy in 2006 and the European Molecular Biology Organization, ENBO, in 2009. In 2015, he was awarded the RDS Irish Times Boyle Medal, Ireland's highest scientific honor. His other recognitions, many of them, include the Wellcome Trust International Prize Fellowship, the B.A. Charles Darwin Award, the GlaxoSmithKline Award of the Biochemical Society UK, and the International Cell Death Society Career Award. Besides a long list of original and review articles, Seamus co-authored the last 3 editions of the major immunology textbook, Essential Immunology. Seamus is currently the editor-in-chief of the FABS Journal, a leading life sciences journal, and the former president of the European Cell Death Organization, ECDO. On a personal note, Seamus has a unique way of balancing his intense scientific work with leisure. You might not guess it, but he is an avid fisherman who finds his active meditation in spending hours near the water, even, or in his case, especially in the coldest Irish weather. This is a testament to his ability to find focus in all aspects of life. That's it. And this is what I can tell, what I can speak loud here.
Right. Thanks a lot. Will I share my screen now?
Share, please.
Okay. And just before I start, how long do I have? How long do I have? About 45?
1 hour and 15 or 1 hour and a half. And
so the total, we have a slot of 2 hours.
2 hours.
You know, no more than that. But with the questions and everything, that normally takes 30 minutes. So
okay.
So yeah, whatever.
No, I'll try to work through it as fast as I can. Thanks, Karina, and thanks, Gustavo and Ricardo, again, for organizing this course. So it's very nice to speak again to you. And like I said the last time, it would be nicer even if I was in São Paulo. But
of
maybe.
Course.
Okay, I'll share my slides. Does everybody see that?
Yes.
Okay. Whoops. Oops. I'm sorry. I'm going to have to go back to the beginning. I was just reviewing them there before I started. Okay. So I'm going to give an overview on essentially cell death from a very kind of basic perspective, why we need regulated cell death. I'm not going to go into all of the different modes, but some of the major modes of cell death. And really discuss what I believe is to be the primary reason for having regulated cell death. And I'll say this right at the outset. I believe this is because when cells die, which they need to do in lots of circumstances, we need to avoid the detrimental consequences of cell death, which in the context of the body is predominantly, I think, the detrimental consequence is inflammation. So regulated cell death really occurs because we need to avoid inflammation when cells need to die. And as I'll explain, cells do need to die in many contexts. So I work in Trinity College Dublin, and Trinity is in the centre of Dublin. So we're quite an old university, I think established in 1759. And my lab is right at the back of the campus here in the centre of Dublin. And that's what it looks like as you walk in the front gates of Trinity. And this is my lab. And some of you may know Ronaldo here on the right-hand side, who until recently was in Gustavo's lab in Sao Paulo. And he's doing pretty brilliant. I'm very pleased with him. And he's doing lots of great work at the moment. So as we know, the cell is the basic unit of life. And we have many, many cells in the human body, approximately 10 trillion or thereabouts. And the body is a cellular society. And like all societies, there have to be rules governing the behavior of the individuals within a society. And the basic rules, to put really bluntly, are cells need to do their job. They stay where they live, and there's exceptions to that. And the primary exceptions to that rule is that cells of the immune system get permission to patrol the body and go into tissues under certain circumstances. But most of the cells basically stay where they're born, and if they don't, then there's detrimental consequences of that. And the primary means of dealing with a cell that moves to a location where it shouldn't be is that it's killed. Another key rule, which again intuitively all know, is that within any individual organism, cells are not allowed to eat their neighbors. They can eat anything that comes from the outside, but they're not allowed to eat themselves, eat each other effectively. So they have to cooperate. And we have, of course, phagocytic cells within the body, but they're only given permission to eat cells under certain circumstances. And as I'll go on to say a little later in this talk, one of the circumstances under which cells are allowed to eat one another is when cells die by the programmed cell death modality called apoptosis. Another rule, just like in society, is that if you live in a house, you can't just build another house in your garden. And in the same way, cells are not allowed to replicate themselves without permission. And that permission predominantly comes in the context of growth factors. So cells, by default, don't divide. We tend to think of cells as being continuously dividing, but that's not the case. They only divide under very particular circumstances. So that's obviously to regulate the precise number of cells with any given tissue. And that's why obviously we don't continue to grow throughout our lives either. Another rule is that infectious agents, bugs, are not allowed inside cells. If cells do become infected, then again, permission is given to other cells within the body to kill those cells. We'll deal with an example of that later in the talk. And again, the type of death that's initiated when cells are affected is a type of programmed cell death. And then finally, a kind of a catch-all category I've said is don't act weird. And what I mean by that is if cells are expressing abnormal proteins or proteins that the body hasn't seen before, then they're effectively treated as potentially infected or on the way to generating a cancer, a tumor. And again, in those circumstances, those cells typically will be triggered to undergo programmed cell death. So in this complex cellular society, cooperation is essential. Every cell should play its role. And as we'll see, there are certain circumstances under which those kind of rules break down. So what happens in the cellular society if you don't perform your job, if you disobey any of those rules? Well, as we'll go on to see, programmed cell death is the penalty in most cases. There's no second chances here. Typically, what happens when a cell breaks any of the rules that I've just mentioned in the previous slide is that they're basically triggered to undergo programmed cell death. So the context I'm talking about predominantly are, especially in the adult, are cells that are injured, that become infected, that act abnormally. So they start to, for example, express abnormal proteins that haven't been seen before in the body, or they've acquired certain mutations that result in abnormal proteins being expressed, or cells that just become dysfunctional. They no longer do the job that they're supposed to do, or they break the rules in any other way. Typically, programmed cell death is initiated, and we'll go through specifically how that happens mechanistically later on. So programmed cell death is used under a variety of circumstances to eliminate cells with surgical precision. And the cells that are indicated here that are red with very intensely staining nuclei, those cells are actually manifesting changes that we, basically characterize as apoptosis. And as you can see, they're very strikingly different from the neighboring cells there. So the two cells slightly to the left that have a green cytoplasm and a nice large blue nucleus, they're just normal healthy cells. But the two cells to the right are cells undergoing a type of programmed cell death called apoptosis. So there's a tremendous throughput of cells, obviously, from when we're born. Everybody starts off as a single cell zygote, which undergoes very precise cell divisions in order to initially develop into a blastula. And then you start to get differentiation of cells into different lineages. And finally, you end up with an adult organism that has, as I say, approximately 10 trillion cells. And in our lifetime, of course, there's lots of replacement of cells within various tissues for a variety of reasons, because of injury, as I mentioned, or infection, or cellular dysfunction, or simply aging. You have an ongoing replacement turnover of cells and tissue. So as new cells are born to replace those that have either become injured, infected, or aged, you have obviously, you need a kind of a similar number of cells to die. And that's effectively why we have programmed cell death, because of course, all of those factors, injury, infection, cellular dysfunction, and aging, are all very predictable. And so our bodies have evolved to be able to precisely regulate both sides of this equation by producing new cells through cell division as cells age and become infected and injured and so on. So in fact, there's a lot of turnover in tissue, and it's been estimated there's about 1,000 times our body weight of cells produced during our lifetimes. And so there's a tremendous amount of cell division as well as cell death that takes place in our bodies at any given moment. That's just restating what I've said. There's plenty of circumstances during which cells die because of, let's say, pathological circumstances, because they're injured or infected, and so on. But also cells die during development in order to sculpt tissues, and we'll see some examples of this in a moment. Before I get into the detail, I'll just point out something that's, I think, very obvious, but it's has several implications. And so we know that cells that basically suffer the most damage or infection, those that are basically closest to the outside surface of the body, these are the cells that you'd expect basically to undergo the highest rates of cell death. And this tends to be in our epithelial tissues. So obviously cells within our skin, within our lungs, in our gastrointestinal tract. These are all cells very much exposed to the outside. This is the source of much of the damage or the potential for infection. And these are the cells that we see the greatest, or these are the tissues that we see the greatest turnover of cells during our lifetime. And as a result of that, these are the tissues that also exhibit the highest rates of cell death, or sorry, of cell division, to replace all those cells that are being damaged and infected and are dying. And as a result of that fact, those are also the tissues that we see the greatest rates of cancer in our body. So if you think about it, the most common cancers are lung cancers, cancers of the gastrointestinal tract, skin cancers, basically anywhere you have a lot of cell damage occurring, and as a consequence, a lot of cell division, you're more likely to get a tumor developing. So cell death and cell division are delicately balanced in the adult. And that's something actually we didn't really realize until about 30 or 40 years ago, when considering diseases of cell proliferation, where we had imbalances either in the number of cells in the tissue, where either we had too many cells or too few cells. Typically, scientists tend to focus on the cell division side of the equation, because cell death wasn't really considered to be under molecular control at that time. It was considered to be something that happened in a passive, kind of unorganized or unregulated manner. Of course, we know now that that's not the case. Both the cell division side of the equation as well as the cell death side of the equation are under a lot of complex molecular regulation. So the body needs to strike a balance between cell division and cell death in order to maintain cell numbers at a precise level throughout life. So let me get into the context where programmed cell death is used specifically to regulate cell numbers in a variety of settings. So first of all, during development, as basically the organism is growing from a single cell zygote, single cell, to literally billions of cells, clearly there is a lot of complex cellular differentiation and cell specification going on. And this is necessary in order to build various structures, organs, and limbs, and very sophisticated tissues. And during that process, we see programmed cell death used time and time again in order to sculpt the developing tissues into their final form. Later on in the talk, I'll discuss a very well worked out example during the development of the nematode worm Caenorhabdus elegans. This is a very small nematode worm that has only about 1,000 cells, and during development, 131 of those cells die at the same times and same places during development. And in fact, studying that worm was very instrumental in identifying a lot of the genes that regulate programmed cell death. So during development, initially the general theme is that cells are overproduced to form a crude version of the tissue. And that's illustrated here by the crude limb petal on the right-hand side here, the upper right-hand side of the slide. Maybe I'll just turn on the pointer here. See if we can see it. So on the right-hand side here, we can see that the crude limb petal has initially been produced through cell division. And then where the arrows are pointing here, cells die through coordinate induction of programmed cell death in order to form the digits as a result of death of the interdigital cells. And a very similar thing happens during the production of blood vessels and many other hollow tubes in the body, where initially a solid tube of cells is produced, and then the inner part of the tube is triggered to undergo programmed cell death in a process called cavitation in order to hollow out the tube. We also see it in a less obvious way during the development of the nervous system. Many extra neurons, probably double the number that you actually need to produce during development. And then as the neural networks wire up, the neurons that don't find productive connections, they die off through programmed cell death. And similarly, during development of the immune system, we overproduced many extra T cells and B cells. And then the specific cells that we require are subjected to essentially a fitness test. And those that fail the fitness test undergo programmed cell death in order to just select the cells that you need in the developed nervous and immune system. I'll give some examples of that later on. So then when we reach adulthood, we have still a tremendous amount of cell turnover, particularly in those tissues I mentioned earlier that are subject to the most damage or infection. And we see a lot of cellular turnover in the gastrointestinal tract, within the immune system, within the liver, and other organs such as the skin that are exposed to a lot of damage in adult life. And then finally, in the context of infection or DNA damage or other forms of exposure to noxious stimuli, we also see programmed cell death being invoked here in order to eliminate cells that are irreparably damaged or that have been infected with viruses or bacteria. So programmed cell death, the message is programmed cell death is used again and again throughout life in order to eliminate cells that for a variety of reasons. Cells that either aren't damaged, but they're just simply required transiently during development, or cells that suffer damage, through their life cycle. Here's maybe a better illustration of development-related programmed cell death. Again, looking at the development of limbs in a little bit more detail, and you can see here this progressive sculpting of the limb pedal to produce the hand in the adult organism. And there's again the example of cavitation I mentioned a moment ago, where a blood vessel is formed through initially producing a solid block of cells, followed by signals that trigger programmed cell death within the cells in the internal part of the developing vessel. And then finally, as the programmed cell death completes, then the vessel is hollowed out. For those of you who study immunology, I think probably there's a good few of you here. You probably know already quite a lot about T cell selection within the thymus. And it's been estimated that between 95 to 98% of developing T cells are killed in the thymus by programmed cell death for a variety of reasons. One reason is because either they don't productively rearrange their T cell receptors, and so they're not capable of responding to any stimulation whatsoever, and those cells essentially die by neglect. And then you also have cells that are too strongly responding to MHC and peptides, so they're autoreactive effectively, and those cells also undergo programmed cell death through negative selection. And then later in life, T cells are also subject to further tests in the periphery, which fine-tunes basically our T cell repertoire, so that we have T cells that are effectively fit for purpose and are not autoreactive, and also they're not non-reactive either. So there's a tremendous amount of programmed cell death that goes on to essentially shape our developing as well as mature immune system. In the context of cytotoxic T cell killing, we have cells that continuously patrol the body, cytotoxic T cells as well as natural killer cells. And when they recognize an infected cell, then they can kill that infected cell via two major routes, either by engaging cell surface receptors on their surface called death receptors, or through degranulating granules at the target cell that contain cytotoxic enzymes that turn on the programmed cell death in the host cell machinery. So programmed cell death is used very repeatedly in the immune system to kill cell populations. Oops. And when controls on cell death fail, the consequences can be really disastrous. So this is a good example of where there's been an interruption of the normal rate of programmed cell death that takes place in the immune system through deletion of a molecule called FAS, which is a member of the death receptor family that's present on cells. And as a result of that, these animals accumulate many extra T and B cells because FAS is involved in turning on programmed cell death in T and B cells, particularly mature T and B cells throughout life. And at the top here, you can see normal lymph nodes. And at the bottom here, you have lymph nodes in a FAS mutant mouse. And you can see the greatly enlarged lymph nodes as a result of the massive accumulation of extra T cells in these animals. And also the spleen massively enlarged in these animals. And typically within 16 weeks of age, these animals develop autoreactivity that's similar to a human disease called systemic lupus erythematosus. So I've just dealt with some of the contexts under which cells need to die. And now we'll start to look at the mode of cell death that typically is initiated when cells do die by programmed cell death. So I mentioned this earlier, but really prior to the 1980s, which I know for many of you here seems like probably a million years ago, but this is when some of us here were actually undergraduate students studying for our degrees. And at that time, there was a lot of focus, as I mentioned earlier, on how cells divide, but there was really almost no focus on how cells die. Because at that time, cell death was generally considered to be, as I say, unregulated, unprogrammed, and it was typically resembled a car crash where cells, were going about their business, and then at some point they got injured or infected or damaged in some way, and then they essentially just crashed. They fell apart. And it was actually very controversial at that time to suggest that cell death could be regulated or programmed. But we now know that cell death is programmed, as I've said, in many different situations. And I'm going to, again, categorize cell deaths here into three categories here. So the first one, which I'll deal with, it's not completely true that this first category called necrosis is unprogrammed. There are certain types of necrosis that are programmed, but broadly speaking, necrosis occurs typically in situations where you have a very severe injury that occurs to the cell. So, for example, in the context of burn injuries or very high levels of poisons, or if you have a severe blunt trauma where you maybe fall over or get hit by a car, then cells can suffer extreme injury and simply burst open. And that type of cell death is called necrosis. And in that situation, it's typically very rapid. I'll show you a movie in a moment. And cells typically rapidly swell and they burst open and they release their contents. And that's, I would say, the exception in the body that cells die by necrosis. In most cases, cells die by a process that's called apoptosis, and that's the regulated type of cell death, or the most common regulated type of cell death that we see. And in this situation, cells sense an injury or damage or infection, and they have a cell death machinery that's switched on by the cell that's been injured or infected. And the cell effectively coordinates its own death. It's an active participant in that process. And there are a set of proteins that are synthesized by the cell that coordinate the death of the cell in that situation. And this is the most desirable type of cell death in a situation where a cell has to die. As I'll explain, it's better that it dies in this programmed, regulated manner than in the chaotic, unregulated manner of necrosis. And then really a kind of a subroutine of apoptosis is a situation where the cell doesn't quite make the decision to die itself, but that decision is made for it by another cell within the body, which I've just mentioned, the cytotoxic T cell or a natural killer cell, which switches on the cell death machinery in the cell that needs to die. And it does so, as we'll see, through engagement of either death receptors within the cell that needs to die, or by delivering enzymes into that cell. Before I describe really what these different modes of cell death look like and show you some movies, I'll just revisit what I said earlier. Does it really matter how cells die when they die? If we do need cells to die in the context of infection or damage or aging, does it really matter how they die? And well, it does matter because, of course, the average cell has lots of potentially noxious enzymes within them. Usually those enzymes are sequestered within different compartments, such as in lysosomes and mitochondria and within the nucleus and different endosomes in the cell. So cells contain lipases and proteases and carbohydrases, DNases, RNases, and DNA itself is actually a very unwieldy molecule. Obviously, DNA is a huge molecule, and it causes problems if it's released from cells as an intact molecule. So one of the reasons why when cell death occurs that it's important that cells just don't burst open is that you want to minimize the release of those nasty enzymes into the extracellular space, because that could cause damage to neighboring cells. And also it can result in activation of the immune system, as we'll see. So does it matter how cells die? Yes, it does matter. Because if you get this type of necrotic cell death, then that is going to result in more cell death as a result of release of all these nasty enzymes. And also our bodies, our immune systems have been trained to recognize when cells burst open via this quite extreme form of cell death. And that typically triggers an inflammatory response, which is good if the cell death has occurred as a result of an infection, but it's not good if cell death has occurred in a more natural context, such as development, for the immune system to be activated. So it does matter how cells die. A more regulated program mode of cell death that would minimize the release of all of these nasty enzymes would be far preferable. So as I've just said, the immune system responds very differently to the presence of necrotic cells to how the immune system responds to apoptotic cells. And broadly speaking, apoptosis is a non-inflammatory or an anti-inflammatory mode of cell death, whereas necrosis is an inflammatory mode of cell death. Now, I don't have time to really go into the details of this, but Polly Matzinger, a very well-known immunologist, essentially realized this about 25 years ago. She wrote a great hypothesis review article at the time suggesting that the immune system doesn't just discriminate between self and non-self. In fact, the immune system doesn't really discriminate completely between self and non-self. It discriminates more between contexts that are dangerous and contexts that are not dangerous. And she proposed that in situations where cells rupture through necrosis, that they release what she dubbed danger signals. They're now called alarmins or DAMPs, damage-associated necrotic patterns. And she proposed that the presence of molecules released from necrotic cells is a very potent activator of immunity, even if there's not any non-self antigen around. And this has become quite an influential idea, which I think is very well accepted now. And we know that at the release of molecules from necrotic cells, which are called DAMPs, are very potent activators of immunity, along with bona fide components of infectious organisms called PAMPs. So as just said there are two major drivers of activation of the immune system: infection in the form of conserved components of pathogens that are detected by a variety of pathogen sensors, such as toll-like receptors or nod-like receptors, and also molecules released from necrotic cells that are dubbed DAMs. They're also very potent drivers of inflammation. And that's a very important step in immunity. You need to either detect a PAMP or a DAMP in order to activate dendritic cells and local macrophages to ramp up the activation of the immune system. And as we'll see, apoptosis is, I think, predominantly about minimizing or avoiding the release of DAMPs when cells need to die. So in terms of the response of the immune system, of course, if you have a live, healthy cell, there's nothing to worry about. The immune system is not going to be activated. If you have necrotic cells, you're going to have the liberation of a lot of cytotoxic molecules, which are going to cause collateral damage within the vicinity of the necrotic cell, and also release DAMPs. And DAMP release then triggers activation of the immune system. And whereas apoptotic cells, many of the events that take place during apoptosis inactivate DAMPs or minimize the release of DAMPs into the cellular environment. And so that's why I say that apoptosis is largely non-inflammatory or anti-inflammatory. I just want to point out something here because I've mentioned inflammation a few times now, and we've begun to realize that, in fact, in response to a cytotoxic stimulus, there's a continuum of responses, many of which are inflammatory to various degrees. So, for example, if you challenge a cell to a dose of a chemotherapeutic drug that has the potential to kill the cell, but not all cells will die to a given dose of a chemotherapeutic drug, we now know that a fraction of the cells will survive and they will persist. And that's often accompanied by an inflammatory response, where those cells signal the fact that they've been exposed to something that has damaged them. At a higher level of exposure to the drug, to the right-hand side here, we often see also that cells they survive the exposure to the drug, but they become permanently damaged, and they exit the cell cycle, and they undergo what's called premature replicative senescence. And these senescent cells are also known to be quite detrimental in certain contexts, particularly in the context of a tumor, because these senescent cells very often release constitutively very high amounts of wound healing cytokines, which actually can benefit the tumor. And there's a lot of interest at the moment in understanding how the senescence-associated secretory response is regulated. That's this type of damaging inflammatory response. At a higher level of exposure to the cytotoxic stimulus, cells undergo apoptosis, and apoptosis here eliminates the cell completely and shuts down any inflammatory response that would have otherwise occurred. And then at higher level damage again, cells can undergo programmed or non-programmed necrosis and rupture and release DAMPs, which are very potently pro-inflammatory molecules. So we begin to realize that there is this really close interconnectivity between cell death and inflammation, with different types of cell death having different inflammatory consequences for the immune system. I'll just briefly mention DAMPs for a moment in terms of what the precise nature of DAMPs are. Since Matzinger proposed their theory about necrotic cells releasing DAMPs, there's been a lot of search for what are the molecules that serve as the major DAMPs. And there's no real consensus on this now, but certainly what has emerged is that members of the extended IL-1 family probably represent the best examples of DAMPs. We know some molecules like IL-1 alpha, IL-33, IL-36, along with some other molecules that are normally never released from cells, from viable cells. These molecules probably represent some of the most potent DAMPs that have been characterized to date. And I'll just skip over that. That's just a little bit more detail on the IL-1 family. So what I've told you so far is that when cells die by necrosis, they're typically potently pro-inflammatory, and also they cause a lot of collateral damage to neighboring cells as a release of cytotoxic molecules. And of course, the immune system, when it gets activated, it also causes a lot of damage, because when you get an inflammatory response, typically you'll get influx of neutrophils, followed by, of course, macrophages, monocytes, T cells, and so on to the inflammatory site. And this can be quite a destructive process. Neutrophils are pretty heavy-handed cells. They have a battery of proteases that they often release into an inflammatory site, and that can cause a lot of destructive damage. So inflammation is best to be avoided unless it's really necessary in the body. During apoptosis, you avoid the release of DAMPs, because as we'll see, apoptotic cells, they present molecules on their cell surface early in apoptosis that facilitate their recognition and removal by phagocytes before they have an opportunity to rupture and burst open. And that's illustrated by this. So we've known for a long time now that apoptotic cells they display changes on their cell surface that trigger their removal by phagocytic cells, or even by neighboring cells that are not professional phagocytes. And one of those changes that we know is called phosphatidylserine. And I'll mention that a little bit later on, but phosphatidylserine is a lipid that is externalized during apoptosis, and we can measure that by using a lipid binding molecule called annexin 5 that can be used as a probe to detect apoptotic cells. So the features, what do the various modes of cell death look like? As I mentioned, when cells die by necrosis, typically they do this in the context of severe trauma or exposure to high concentrations of toxins, such as alcohol, for example, or extremes of heat or cold, or high doses of radiation. And the sequence of events usually looks like this. Cells that are adherent, they very rapidly round up, they start to swell, and then quickly they burst open and release their contents. And this is what it looks like in the lab. These are cells that have been exposed to high concentration of hydrogen peroxide. This movie was shot over a period of an hour. You can see that there are blebs forming that just burst. And all of the cells in the culture manifest the changes very quickly. And cells go from a state of being viable, perfectly, healthy, to being completely ruptured in the space of an hour or two. And all cells die, you can see here, in a fairly coordinate manner. So that's what necrosis looks like. It's typically swift and rapid and extreme, and results in the release of essentially most of the contents of the cell. When cells die by apoptosis, we typically see that this occurs in much more physiological contexts. And in fact, many of the things that trigger apoptosis don't even damage the cell. Molecules like TNF or FAS or TRAIL or withdrawal of growth factors, they can all induce apoptosis. And in that context, the cell isn't injured in any way, but it's just had receptors engaged on its surface. But also mild forms of injury or damage also activate apoptosis. I'll just, before I, I'll show the movie in a moment. In case you're interested, the first use of the term was a paper published by John Kerr, Andrew Wiley, Alistair Curry in 1972. And this paper introduced the term apoptosis because this group of scientists had realized that they were observing cells that looked abnormal in various situations, where they realized the cells must be undergoing some type of novel cell death that was regulated. And they introduced the term apoptosis for this type of what they proposed to be programmed cell death. They didn't have any proof for that at the time, but that paper was very prescient, and I would strongly advise you to read that paper if you're interested in apoptosis, because it has lots of beautiful electron micrographs of cells undergoing apoptosis and also apoptotic cells engulfed within neighboring cells and macrophages. And they made a lot of predictions in that paper that subsequently were borne out to be true. And they introduced the term apoptosis because they wanted to parallel the use of the term mitosis, because they felt that this was the opposite to mitosis, but it was also regulated just the way mitosis was. And in that paper, they likened the process of apoptosis to similar to how a tree loses its leaves in autumn, where that's just a natural part of the life cycle of the organism, and wasn't reflecting any particular catastrophic event, but something that occurred quite naturally and quite normal in a regulated manner. So this is what apoptotic cells look like. On the top here, we have viable, healthy cells. The nucleus has been stained here by DAPI in blue. And then when cells are undergoing apoptosis, you'll notice that the nuclei have become fragmented and highly compacted. And that's a very characteristic sign of apoptotic cells, this nuclear morphology. And then at the bottom here, we have cells undergoing necrosis, where you can see that changes have happened to the nuclei, but they haven't become compacted and basically broken up into small pieces like we see during apoptosis. And in fact, the nuclear compaction and fragmentation was the first morphological characteristic of apoptotic cells that was identified in the original Kerr et al. Paper. And this happens for a number of reasons, but one of the reasons because a very striking hydrolysis of the DNA occurs during apoptosis, and that's the hydrolysis of DNA into 200 base pair multiples as a result of activation of an endonuclease in apoptotic cells, which hydrolyzes the DNA at internucleosomal sites, essentially at any unprotected sites in the genome. This nuclease called CAD, it becomes activated, and it then hydrolyzes the DNA. So in apoptosis, what happens is cells typically detach from the substratum, they round up, the nucleus condenses and then starts to fragment into these highly condensed pieces. The plasma membrane usually undergoes a fairly extensive blebbing. And then finally, the cell can start to break up into intact vesicles. And this is probably to facilitate uptake of the cell by phagocytes to make it a bit easier. And typically, these cells then still remain viable. They don't leak contents. They don't burst open. They remain viable for about 24 hours. But even at the earliest stages, these cells will have displayed alterations to their plasma membrane that will enable neighbors or phagocytes to recognize them as being in the throes of apoptosis, and will trigger their engulfment and removal before they can burst open. So, and also the process of apoptosis is much more drawn out. It typically takes place over a period of 12 to 24 hours, unlike necrosis, which takes place in about an hour. This is an example here, a scanning electron micrograph of an apoptotic cell surrounded by neighboring cells. You can see that all of the little membrane apoptotic blebs that are being produced. And this cell would have externalized this membrane phospholipid phosphatidylserine that is not normally present on the surface of viable cells, but in apoptotic cells, it's externalized very early. This is what the process looks like. In this case, we've cells, I think, exposed to cisplatin or daunorubicin. And this movie is shot over a 24-hour period. And you can see that cells independently undergo apoptosis at different times. And there are some cells within the culture that really take a long time to initiate the process. And yeah, so in fact, many times we do these experiments, we can choose drug doses that will only kill maybe 40 or 50% of the cells, and the remaining cells completely survive and go about their business. And that's because the cell is participating in the decision-making process by essentially making a molecular decision to switch on its cell death machinery or not. And we can block this also by either knocking out or inhibiting some of the enzymes involved, which I'll get to, are called caspases. And these are proteases that play an important role in coordinating this type of cell death. And again, I'll return to the fact that apoptotic cells are very rapidly recognized and removed by phagocytes early in the process. So they take many hours to manifest the apoptotic changes, but from a phagocyte's point of view, it can recognize apoptotic cells within an hour or two of switching on its cell death machinery. And this avoids the release of the contents of the apoptotic cell. And so the cell is engulfed and digested in the macrophage safely. And so you avoid the release of DAMPs and the minimization of inflammation. This is what I've said a few times now, that one of the signals that triggers the macrophages to eat apoptotic cells is the externalization of this lipid called phosphatidylserine. And there are other molecules that act to attract phagocytes to apoptotic cells. They're called find me signals. And annexin-5 is a lipid binding protein which can be used to detect apoptotic cells by labeling with FITC or another fluorophore. And that's an example here of annexin-5 labeling of cells. And the left-hand side here, we see this is healthy cells. And then within a couple of hours of triggering apoptosis, we have a population that are binding annexin-5 quite avidly. And then within a couple more hours, we have the majority of the population binding annexin-5. And then we also, as a population up here, that are now starting to leak. So they're binding annexin-5, but also they're starting to leak after quite a number of hours. I've said this multiple times now just to reinforce the message, but in essence, the major difference between apoptosis and necrosis is that necrotic cells burst open, they release DAMPs that triggers activation of DCs and macrophages, and you get an inflammatory response, whereas apoptotic cells, minimizing that occurring because they effectively package themselves into a kind of a phagocytosable particle. They display signals that trigger their engulfment by phagocytes, and as a result, you don't get an inflammatory response. And in fact, it's even been shown that upon engulfment of apoptotic cells by macrophages, they switch into an anti-inflammatory mode, although that can be context dependent. Okay, so I've dealt with necrosis, what it looks like, and apoptosis, what it looks like. And then we'll turn to this other type of apoptosis where the apoptosis is switched on in the cell by a cytotoxic T cell or a natural killer cell. And it looks like normal apoptosis, but it is a slightly different category because the cell that's dying doesn't make the decision. The decision is made for it by the cytotoxic T cell or natural killer cell, delivering either a death ligand to the cell surface or a cytotoxic enzyme within the target cell. So I showed this earlier, and we'll return to it a little bit later on. So again, in culture, the cells moving around are the, in this case, natural killer cells. And you can see here on the left, this cell here, this NK cell, is going to recognize this cell, and it's going to degranulate, and it's going to kill that cell, and you'll see that cell undergoing apoptosis. So the cell is degranulating now, it's activated, and this cell is going to undergo apoptosis as a result of that. So during the granulation, the natural killer cell delivers an enzyme called granzyme B into the target cell, and granzyme B is a very good way of switching on the program's cell death machinery within cells that are infected or behaving abnormally in some other way. Okay, let's look at the molecular mechanism. I'm going to go through this quickly in the interest of time. So how do cells die by apoptosis? What's the molecular machinery? How does it all work? Well, an important organism for working out the genetics of programmed cell death has proved to be the nematode worms Xenorhabdus elegans. This worm was basically introduced to the community by Sidney Brenner, working at the LMB in Cambridge in the 1960s, who wanted a model organism that was complicated, fairly big. So more complicated, obviously, than yeast, for example, but not too complicated. So not as complicated as a fly. And he chose Xenorhabdus, initially Xenorhabdus briggsei, but then switched later to Xenorhabdus elegans, because it was an organism that he felt that you could do genetic mutagenesis studies in, that was quite small, you could propagate it easily in the lab and do genetics in this organism. And it proved to be a very good decision. And of course, there's a huge C. elegans community now that are using C. elegans in order to do genetic screens of a lot of different processes. And this is what C. elegans looks like from development from a single cell. You can see now very quickly it has hundreds of cells. And this takes place within a few hours. And then the worm starts to take something approaching a worm form. As you can see here, it's moving around in the egg case. Now it's an actual kind of proper worm. But we've seen it go from a single cell to having 1,090 cells in order to form an adult worm. But of those 1,090 cells that are born during development in the worm, what they noticed when they study these worms under Nomarski microscopy is that they noticed that of those 1,090 cells that were born, 131 of those cells always died at various times during development. And at that time in Sidney Brenner's lab, there were two, what have now become very well-known molecular biologists, geneticists. On the right here, we have Bob Horvitz, and who's not pictured as John Solston. They were both postdocs in Sidney Brenner's lab at the time. And Brenner basically put them to doing genetic screens on the worm to find genes that were involved in regulating these 131 cells that died during development. And these screens were very successful, and they identified a series of mutants, which they called SED mutants, cell death defective mutants. And there were, so they called them SED1, SED2, SED3, SED4, and so on. And I think they identified about 11 SED genes. And these mutants fell into categories where either the worms had either too little cell death, so none of the 131 cells died, or a fraction of the 131 cells died, or worms where there was too much cell death. And then they also had worms where the cell deaths were normal, but the dead cells were not recognized and engulfed as they normally were. And, over a period of a decade or so, in fact, a little bit more than that, they then cloned those genes and started to look at their function and how they interacted with one another. And this is the paradigm that they established. This is a kind of a blueprint for cell death in the worm. They found that SED3 and SED4 drove worm cell death. They were essential. If you knocked out SED3 or SED4, you got none of this programmed cell death that took place during development. And then SED9 normally blocked cell death. So if you overexpress SED9, or you had mutant worms that had gain of function mutations in SED9, you also blocked cell death. If you lost SED9, you got catastrophic cell death in the worm. And then another regulator, which came out in another screen initially, called EGL1 or egg-laying 1 defective. And that was an inhibitor of SED9. And as we'll see, it turns out that that's a very good blueprint for how programmed cell death also works in mammals. We have the homologs of SED3, which are the caspases. We have a major homolog of SED4, which is called APAF1. And then we have homologs of SED9, which are called the BCL2 family, and the homologs of EGL1, which are the BH3 only proteins within the BCL2 family. So I'm going to give a bit of a whistle-stop tour of how this works as we go from C. elegans to Drosophila to humans. But in essence, we're going to see the same platform is used again and again. And that's a platform between SED3 and SED4. So SED4 is essentially an activation platform for SED3. Normally, SED3 is inactive in healthy cells. And in order to get programmed cell death, you need to activate SED3. And you do this by liberating SED4 from SED9. And this is achieved through EGL1 displacing SED9 from SED4. SED4 oligomerizes into this activation platform called the apoptosome. And then it recruits SED3. And then that results in SED3 activation. And then SED3 being a protease, it cleaves lots of proteins in order to coordinate programmed cell death in the worm. And what we see in Drosophila, and I should mention, of course, Bob Horwitz and John Solston and Sidney Brenner received a Nobel Prize for their work on programmed cell death in the worm. They received that in 2002. In Drosophila, it's similar but different. Similarities are that now we have multiple SED3 homologs that drive programmed cell death in the fly. We have a SED4 homolog called DARK or Drosophila APAF1 related killer, which activates the SED3 homolog drunk. And it's a little bit different because the SED9 homologs are not important in the fly. They're there, but they don't play a role in programmed cell death. Instead, the fly has worked out its own kind of solution to activating the apoptosome, and that's through molecules called Reaper, Hidden, Grim. They're also present in mammals, but they don't have a really important role in driving cell death. But what they do in the fly is they neutralize this inhibitor of apoptosis protein that can block the activity of the drunk caspase. And so the wiring is a bit different here, but in essence, you ultimately get the formation of an apoptosome, again, in the fly, comprised of DARK and drunk. So again, DARK is a APAF1 or a SED4 homolog. Drunk is the SED3 homolog. And, you know, the regulation is a bit different, but the overall downstream complex is quite similar. And then when drunk gets activated, it triggers activation of other fly caspases, proteases, and then they go on to coordinate programmed cell death. And in mammals, it's also similar but different. We have the SED3 homologs called the caspases. Of course, it's now become more elaborated as a result of gene duplication and divergence. You have multiple caspases involved, not just SED3. You have APAF1, which is involved in activating one of those caspases, which we'll see. And then you have the SED9 homolog, the pro-survival members of the BCL2 family, and then the EGL1 homologs, the BH3 only proteins. And there's some other bells and whistles in mammals, but effectively you have the same kind of blueprint for programmed cell death in mammals, where you have a whole slew of SED9 homologs. And we also have IAPs, just like the fly has, and they play a kind of a modifying influence in regulation of cell death in mammals. They're important, but they're not that important as they are in the fly, but they are present. And then we have a whole bunch of SED3 homologs called caspases. And we have a similar formation of a apoptosome complex comprised of caspase 9, APAF1. And so much about getting programmed cell death in mammals is about activating this complex. And there are three major routes to activating the mammalian apoptosome. One of the routes, which is called the intrinsic pathway, is through activation of the BH3 only proteins. I don't have time to go into how this occurs, but basically diverse forms of cell stress or damage activates one or more BH3 only proteins, and then they activate other members of the BCL2 family called the back channel, That results in cytochrome C release in mitochondria, and then that triggers assembly of the apoptosome, which activates caspase 9, and you get a slew of other caspase activation events. So there's another major pathway, and that's through death receptors, which neither the fly or the worm have. These are cell surface receptors that, when engaged, they activate another caspase, caspase 8, and that can activate a BH3 only protein called BID, and then BID can permeabilize mitochondria and again activate the apoptosome pathway. And then the cytotoxic T cell and NK cells I mentioned earlier, they deliver granzyme B into the target cell, and granzyme B is a protease that can also process and activate caspase 8 or BID and engage a pathway similar to this death receptor extrinsic pathway. So we have three major pathways that have been identified in mammals, but we still have downstream, we've got this apoptosome and caspase activation cascade. And then ultimately caspases coordinate cell death by cleaving thousands of proteins within the cell that produces the apoptotic phenotype. I know I've gone through this pretty quickly. I hope you've got the basic thrust of it, but there's lots of reviews out there on this topic. And, you know, for those of you who are interested about the details, there's lots of reading that you can do. I'm going to skip that. I just really want to mention that in terms of how caspases kill, as I've said now, they cleave lots of proteins in the cell. We know the consequences of many of these cleavage events. Earlier, I told you that DNA becomes hydrolyzed into 200 base pair multiples in apoptosis. And this is achieved through hydrolysis, caspase-dependent hydrolysis of this inhibitor of DNAs called CAD. So CAD is normally kept inactive by its inhibitor ICAD. And so when ICAD is cleaved by caspase 3, that activates CAD, which is depicted here as an arrow, and then it can then hydrolyze DNA. There's lots of other caspase substrates that have been identified that are cytoskeletal proteins, so lamins, fogin, vimentin, actin, and that effectively dismantles the cell and starts the blebbing that occurs during apoptosis. And there's quite a few other important caspase substrates that have been identified. Again, we've summarized this in a review a number of years ago in Nature Reviews Molecular Cell Biology of how caspases effectively coordinate the demolition of the cell through cleavage of strategic substrate proteins within the cell, and that's what collectively produces the apoptotic phenotype. I think I'm going to skip a lot of this. I just want to make an important point here. This opening of a mitochondrial pore to release cytochrome C is a very important step in apoptosis in mammals that we don't have in the fly or in the worm. And this is really what the BCL2 family do in mammals. They essentially regulate the opening of this back channel, and that's a very important step in apoptosis, the opening of that channel. So once back get oligomerized, they permeabilize the mitochondrial outer membrane and release cytochrome C. And as I mentioned earlier, this is how the apoptosome gets activated in mammals. And that's normally restrained by the pro-survival members of the BCL2 family, such as BCL2 and BCLX, MCL1, and so on. So we have the same genes involved as we have in the fly and the worm, but they function somewhat differently. So in the worm, the SED9 family, the SED9-AGL1 players, they directly regulate the apoptosome. But here in mammals, we have the SED9 homologs, the BCL2 family, and the AGL1 homologs, the BH3 proteins. They don't directly regulate the apoptosome by binding to it, but they instead regulate the release of a factor from mitochondria that is required for apoptosome activation. That factor is cytochrome C. And there's been a lot of investigation into that over the years. I think I'm going to just finish up by mentioning that, as I mentioned at the outset, when apoptosis goes wrong, either you have too little apoptosis or too much apoptosis, then inevitably disease results. So we know that in cancer, cancer is not just a disease of overproliferation of cells, it's also a disease of too little programmed cell death within certain cell populations. So you can get cancer by either having too much division or too little cell death. And many other diseases, such as neurodegenerative diseases, occur as a result of too much cell death happening as a result of cells inappropriately undergoing apoptosis in different circumstances. I'll just go back up one slide and say something I've said now lots of times. The overarching point I want to make is that we have programmed cell death in order to control the response or the consequences to when cells die. Cells are going to die in lots of circumstances in the body. That's unavoidable. But what is avoidable is the inflammatory consequences. If cells die by necrosis, then typically this is a context where you're going to get strong inflammation. And if cells die by apoptosis, then typically what happens is the cell is recognized and removed from the tissue before it can burst open and release inflammatory drivers. And so that's typically non-inflammatory mode of cell death. And so that in a nutshell, I think is the purpose of programmed cell death, to regulate the inflammatory consequences of cell death in the body. And I'll leave you with just a note that although we've been working on cell death for 30 years, we know, of course, most of the major players in the process. There's still a lot to do. Cell death research is not dead. There's still lots, I think, that is remain to be discovered. And something I'm quite excited about at the moment is thinking about and working on how the decision between the different outcomes is made when you challenge a cell, for example, with a chemotherapeutic drug. If you treat cells with a dose of that drug, that is the typical dose, for example, a patient would receive, then you'll find that about 20% of the cells undergo apoptosis and 50 to 60% of the cells enter senescence. They don't die, they survive. And a fraction of the cells basically transiently arrest, and then they re-enter the cell cycle and they continue to divide. And of course, that's actually what happens in the therapy situation. We know that during chemotherapy, patients are not given just one dose of drug and then their tumors are cured. They have to be given multiple doses of drug, usually, weekly cycles over a period of 3 months. And so in a lot of cases, you're having this outcome here or this outcome rather than the desired outcome, which is to kill the tumor cell. And I think one of the kind of very understudied and unexplored areas right now is identifying the molecules that kind of regulate the checkpoint or the decision between undergoing senescence or apoptosis or indeed persistence of cells. And so I think we're still at a reasonably early stage in cell death research. And finally, just a note on why to do research. If any of you here are considering research, it's a great thing to do as a career. There's a saying, if you find something you love, you'll never work another day in your life. And, research feels like that at times. Not the grant writing bit, but, the actual looking down the microscope bit. It is fascinating to work on something that is complex and beautiful and also very relevant to health. And, it's a very enjoyable career. So I'll finish up by a little plug for Essential Immunology. If you're buying an immunology textbook, that's a great one to buy. So thank you.
All right, man. Thanks a lot for your brilliant talk. I think Karina will manage the questions. There's a lot of questions in the chat already, but I will ask people to open the mic and then ask the questions themselves.
That's it. So thanks, Shay. Brilliant as usual. And now I think Igor was the first student to ask a question. Igor,
Are you listening?
where are you? Yes. Could you
Uh,
open your camera, please?
I'm on the computer and I don't have webcam.
Okay, so do
Great
your question.
presentation, Professor. And which is the most common cell death in an infection, induced by immune system or suicide by the cell? By the cell.
I think in the context of infection, early in infection, I suspect that depending on the nature of the infectious agent, I suspect there's a degree of necrosis early in infection. But then, as of course, cytotoxic T cells and NK cells come in, much of the death will be by programmed cell death, by apoptosis. And also remember, particularly viruses encode molecules that can also disturb or interfere with the activation of the cell death machinery. So it can be made complicated as a result of that. If the viruses encode inhibitors of caspases, which many of them do, or inhibitors of other molecules that are involved in the regulation of cell death, then you can get complex outcomes. So it really depends, I think, on um, the titer of infectious agent. If you have very high concentrations of virus, I think you can have different outcomes in that situation. The nature of the virus, or indeed pathogen itself, or bacterium. So it's hard to give a very clear answer to that. It's context dependent, really. But you will have lots of apoptosis going on, but you will also have a form of cell death, which has been termed necroptosis, which is where apoptosis is effectively frustrated in the cell as a result of virally encoded proteins blocking the caspase activity. Then there seems to be a default pathway which can be activated by the cell, which Peter has done a lot of work on. And that's called necroptosis, where if the virus interferes with caspase activity, then you can get activation of RIPK3 and MLK1, and that can form a pore to basically drive a type of programmed necrosis. So I mentioned a couple of times that there are forms of programmed necrosis. Another important form of programmed necrosis called pyroptosis, also in certain infectious settings. And that can result in a programmed mode of necrosis, again driven by another pore-forming protein there called gasdermin D. So in infectious settings, you can get necroptosis, pyroptosis, apoptosis, or direct necrosis, let's say, at very high titers. So it's hard to give you a simple answer, unfortunately.
That's it. And Lucas, sorry.
Oi.
God, I was gonna say that they can, just raise the hand with the reaction.
Oh, I think it's better.
Yeah. So just raise your hand and then open up the mic
Yes.
and then start. So we don't need to keep, calling you guys. All right.
So, but Lucas, please move on.
Okay. I asked you one question that was answered during the presentation,
That's it.
but it was based. The question was if it's correct to characterize necrosis as inflammatory cell death and apoptosis as non-inflammatory cell death. And I asked this question because I know that necrosis, there is a release of intracellular factors and promote inflammatory response, but apoptosis, there is markers of inflammatory, and it's also related to healing of the tissue. And why it's not inflammatory cell death? Because we use markers, and macrophage is also of the immune system, one kind of the cells of the immune system. I don't know if correct to talk in this way, but I think that there is a immunologic process involved in apoptosis too.
Okay. When you say there's markers of inflammation and apoptosis, which ones are you thinking of?
I am thinking, I'm not of the immunology department, but I am studying in neuroinflammation. And if I induce apoptosis in the brain, uh, I expect that we will have a microglia activation and increase of astrocytes in the tissue because of the healing. And it's not one inflammatory process, neuroinflammatory process.
And the context you're thinking where you say if you induce um, cell death in the brain, how would you induce cell death in that situation?
With NMDA agonist.
Yeah. So you're talking about, I mean, look, so nothing is binary. And as I said, I think apoptosis minimizes inflammation. It's not to say you won't have any inflammation where you get apoptosis, but it's about minimization of that inflammation. I think in the situation you're talking about is you can't exclude the possibility that neurons would die by necrosis in that situation as well. But I think it's a question of, in biology, everything is about, the magnitude of the effect or the stimulus you're talking about. So if you have a lot of apoptosis, then you are going to get inflammation in that situation, partly because the local phagocytes are not going to be able to cope with the sheer scale of the apoptosis going on, and their phagocytic capacity will be exceeded. And so inevitably, some of the apoptotic cells will undergo what's called secondary necrosis, and they will burst open and you will get inflammation there. So in an in vivo setting where you're using a kind of a bolus of, a compound to trigger an abnormal amount of apoptosis, I think you are going to get some necrosis, and also you're going to overwhelm the capacity of the phagocytes to deal with the apoptosis there because it's much greater than normal. Whereas I'm talking about settings where apoptosis occurs more naturally, and in those settings, typically apoptosis occurs in isolated, scattered cells that would not get anywhere near close to overwhelming the phagocytic capacity of the tissue in that situation. For example, if you look at the liver, where we know there's a tremendous amount of apoptosis going on at any given time, or the gastrointestinal tract, you don't see any inflammation going on there, and you rarely see apoptotic cells. In fact, if people have done these counts where they've looked for apoptotic cells in the liver, at any given time, the numbers are like 1% or less because they're so efficiently recognized and eliminated. So my answer really is, okay, if you use a stimulus that greatly elevates the amount of apoptosis in the tissue, you can probably overwhelm the capacity of the tissue to cope. And also you may trigger some necrosis in that situation. But apoptosis, broadly speaking, I think absolutely is a non-inflammatory mode of cell death. And for example, if you produced an infarct in the brain by a more, aggressive stimulus and produced, necrotic cell death, I think you would see far greater evidence of glial cell activation and inflammation under those situations. So the point I'm making is not that apoptosis is absolutely completely non-inflammatory, but it is far less inflammatory than the alternative, which is necrosis.
Thank you. But I wanted to ask another thing. If I am studying neuroinflammation in neurodegenerative disease, and I expect to find apoptosis, I can talk about neuroinflammation.
You say that again. You expect to find apoptosis?
If one neurodegenerative disease induce apoptosis, I can talk about neuroinflammation.
You're asking me, can you talk about neuroinflammation?
Yeah, this is a question.
If you have evidence of inflammation, I guess, but don't ask me in that situation.
No,
Yeah.
But during the neurodegenerative disease, you can have pyroptosis and necroptosis instead of apoptosis, of course, or in addition to apoptosis. So because of this form of cell death, you can have neuroinflammation.
I think
Okay.
it depends on the trigger of the neurodegeneration. If you have large aggregates of amyloids, in those situations, cells may well die by necrosis. And as Karina mentioned, I mean,
Apoptosis.
I have to say, I feel the whole neurodegenerative area is a bit of a minefield. I'm not sure that people have still got to grips with exactly the modes of cell death that are actually taking place in a living brain. You know, a lot of the work that's been done on neural cell death has been done obviously in vitro still. But the brain is a little bit of a, I would say, a complicated example. So,
But
Okay, thank you.
sorry, can I jump in the discussion? Because another thing is that what do you consider inflammation? Okay, so Seamus mentioned the sculpture of organs, of limbs. Okay, so during the cell death, the massive cell death that you observe in this situation, you have a huge accumulation of macrophages, which will eaten up the dead cells. So would that be considered inflammation, this migration and accumulation of macrophages? So I think that's what you should measure. What sort of what are the evidences of inflammation? Just
Inflammation.
cell migration or something else?
Inflammatory cytokines or
yeah, inflammatory cytokines, I think.
Exactly. It was because of this question. Thank you.
Guilherme.
Hi, Ka. Hi, Gustavo. Just a
Thank you.
quick follow-up around on this same topic of discussion. Can you guys hear me well?
Yep.
Yes.
Yeah, okay, perfect. So even if you have like evidence of having apoptosis, apoptotic death in the cells in the nervous system, if you don't have the immunoclearance of it, it could also trigger like secondary apoptosis after a while. And then I believe that maybe this secondary apoptosis will have like a more pro-inflammatory reaction in there, and maybe even make
Yeah.
like neighboring cells succumb to necroptosis or more like necrotic like phenotype deaths in there. Is that correct to assume, or I'm just tripping over my mind here?
Yeah, as I mentioned, you know, it's a question of scale. So if you have, you know, low amounts of apoptosis going on, you know, stochastically, then the tissue will cope. If they're greatly elevated, then you may exceed the capacity of the tissue to remove them. And then in that situation, the cells may undergo secondary necrosis, it's called. But they may have primarily died by apoptosis. And then if they're sitting around long enough, you know, they will burst open and release their contents.
Oh, okay.
That is effectively, you know, necrosis. I mean, some of the dams have been inactivated as a result of caspase mediated attack, but yeah, I think it's like the case of the infection scenario. It's going to be context dependent. It's a question of how the damage in the brain has occurred. You know, what's the stimulus? You know, what's the degree of damage? You can't generalize too much in these situations.
Yeah, almost will depend on the trigger itself, right?
I think it will
What
depend on that
started the whole, cause yeah.
and the degree of
Okay,
damage.
thank you. Thank you.
Thanks.
Letícia Savério.
Can you hear me?
Yes.
Yep.
Okay. So great presentation, Professor. It's pretty cool. My question is thinking about a neuron or a female X in the ovary. We already know that cells has a limited proliferation and certain resistance to cell death. So could you say which cell death mechanisms for these cells are more robust to guarantee a longer life of the cells in compared to other cells in our body that go to autophagy more frequently the female eggs or neurons. In the end of your presentation, you mentioned some proteins that control cell death. Could you mention more about it?
Yeah, I mean, we do know that different tissues have different thresholds for entering apoptosis or other endpoints upon damage. So I did mention at the beginning of the talk that we know in epithelial tissues, there's a very high rate of cell turnover. Those tissues are typically damaged, much more frequently than other tissues, such as the brain. And as a result, they tend to have a greater propensity to enter apoptosis. Epithelial tissues are usually typically easy to kill. And because they're easily replaceable generally, they're not very specialized tissues. Complex tissues, such as the brain, especially where the experience of the cell is kind of impacting on its function. So if you have a highly innervated neuron, that experience is very costly to throw away. And those cells typically, I think they have a much higher threshold for killing them. And so they will have alternative mechanisms to spare the life of the cell. So DNA repair, autophagy, I think, yeah. So for example, we know in lymphocytes, DNA repair is possible, but is probably never really mounted in lymphocytes. It's very easy to replace a lymphocyte. So the outcome, even in response to very low doses of DNA damage, is usually apoptosis. Whereas in other long-lived tissues, the outcome is probably much more likely going to be DNA repair and survival of the cell. So bottom line is, in more specialized long-lived tissues, I think the survival mechanisms, repair, are going to be invoked, but in less specialized tissues, then typically the threshold for death is very low. And if a cell is damaged, it will activate its programmed cell death machinery. So clearly the repair mechanisms we're talking about are autophagy, the unfolded protein response, the DNA damage response, the heat shock response, the reactive oxygen handling response. There's a whole variety of stress response pathways that can be invoked. But whether they're effective will, I think, depend on the context. So if you think about it, cardiac tissue is, low turnover, so is skeletal muscle, so is brain tissue. Those tissues are probably much more reliant on the repair mechanisms when they're faced with the stress. And they probably have a very high threshold for entering cell death. But the epithelial tissues that are subject to the most damage and they're less specialized, those tissues probably don't mount very effective repair responses in most cases. The cells simply die and they're replaced through the vision of a neighbor. So I think the repair versus death responses is going to be different in different tissues. There's a nice study actually about a year ago from Chris Sarasak's lab, published, I think, in Nature Communications, where in fact they looked at this quite precisely in a lot of different primary cell types. And they found that, that the kind of threshold for activating this mitochondrial pathway where BAX and BAC channel opens, they found that threshold is markedly different in different primary cell types, which supports the idea that, there are vastly different thresholds for engaging apoptosis depending on the tissue type.
Awesome. Thank you.
G.
Yep.
Hello. So, in your opinion, what do you think is the best technique to assess apoptosis?
Well, it really depends, again, whether you're looking in vivo or looking in vitro. I think the best technique is actually to use morphological criteria, look down the microscope and know what an apoptotic cell looks like. That's what we use in our lab most of the time. And then we support that by then using other, indicators of apoptosis, such as staining the nuclei, measuring caspase activity, the cleavage of caspase substrates. It's kind of like saying, what's the best method for identifying a dog? Your eyes. Okay. You can start adding other characteristics if you like, but you know what I mean? When you know what a dog looks like or a duck, you can tell. And we measure apoptosis predominantly by looking at the microscope. I can tell the difference between necrotic cells or apoptotic cells very readily. If we get an alternative kind of strange outcome, then we start to look at molecular markers. But if you want to prove it in the context of a paper, then things like staining for nuclear condensation and breakup of the nucleus, that's very characteristic of apoptosis. You very rarely in any other context get this hypercondensation of the nucleus and breaking of the nucleus into many different fragments. You can look for caspase activation by Western blotting or by using caspase substrates such as VAD, then labeled with different moieties like FLICA. It depends. It really depends on the context you're working in. Have you got a particular context in mind?
More in vitro, like to differentiate between deaths, because like you said that apoptosis is really characteristic, right?
It is.
Yeah.
And normally, under a given circumstance, you'll just usually get one mode of cell death at a given threshold of stimulus. It's usually one, you won't see usually mixed apoptosis and necrosis in a culture normally. If you treat cells with a drug or another stimulus, if the cells are going to die, they'll typically,
Choose one. Okay.
They'll typically enter apoptosis, let's say, at a certain dose of the stimulus. You'd have to really raise the stimulus level up very high to get necrosis. So there, it's kind of fairly bimodal in that respect.
Okay, thank you.
All right.
So students, please again look at the cells. So dog or duck?
Yes.
Vitor?
Hi, great talk, Professor. It's me, or there's another Vitor?
Yes, yeah.
Okay. I have a question about an image that you showed before about the flow cytometry. That we usually use flow cytometry to see cell death by annexin 5 and PI. And in that image, I saw that the annexin 5 and PI positive events were named as secondary necrosis. But I also see that some works named these annexin 5 and PI positive events as late apoptosis. So it's wrong to call these events as late apoptosis, or
no? I mean, it is late apoptosis. Yeah, I mean,
I don't know the difference.
The difference is just that I'm calling it secondary necrosis, which is widely used in the field, where the apoptotic cells start to leak open. If they're sitting around long enough, they start to leak and they start to take up vital dyes such as propionate iodide. It's a nomenclature distinction. You can call them late apoptotic cells as well. That's what they are. Yeah, they're late apoptotic cells. But functionally, they've become necrotic cells now because they're releasing their contents. So they would behave, in terms of their functional impact, quite like necrotic cells. But yeah, late apoptosis or secondary necrosis. Either term is fine.
Okay, thank you.
Okay.
Isabella?
Hello, can you guys hear me?
Yes.
No, not at all.
So let me just read the question I typed here. So Professor, first of all, thank you very much for this engrossing lecture. I wanted to ask you, in your opinion, when it comes to solid tumors, do you think that necrosis can play an important role in killing tumor cells while activating the immune system, in comparison to apoptosis that can be immunosuppressive?
Again, I think a tumor is a situation where you have everything going on. So we know that tumors, once they reach a certain size and they start to become hypoxic, they develop a necrotic core. So they literally suffer from oxygen deprivation if they're not sufficiently vascularized. So in that case, the tumor will experience oxygen deprivation, nutrient deprivation, and cells will die by necrosis. We know that. That's, all tumors beyond, 1 or 2 centimeters cubed develop a necrotic core. I think that's an important driver of inflammation, because you'll have a lot of DAMPs released in that situation. That's going to trigger ingress of inflammatory cells into the tumor. And then you're going to have cells within the tumor also dying by apoptosis, in that they won't have experienced this severe oxygen or nutrient deprivation that you get in the core. But those cells are clearly under stress. They're growing in a place they shouldn't be. They will also experience clearly nutritional deprivation. And there's undoubtedly going to be apoptotic cells in tumor as well. In terms of whether you're saying the apoptosis could be, what was the term you mentioned? I just forget.
Immunosuppressive.
Yeah, I don't think so. I think probably what happens generally is that, regardless of the mode of cell death, once you get recruitment of inflammatory cells into a tumor, the question then becomes, okay, what are those? What if they start becoming activated as a result of DAMPs or other stimuli within the tumor, and you have local T cells that now are capable of being primed by those dendritic cells? What are the T cells going to recognize? What's the tumor antigen? So in most cases, I think what will happen is strong tumor antigens, it will be cells that bear them will be deleted. And then you will end up leaving subpopulations that don't have strong tumor antigens. And so you'll get essentially any T cells there will effectively be tolerized. And I think, because tumors are, remember, they evolve over a very long period of time in a Darwinian process. By definition, if you have a strong tumor antigen, that cell is going to be eradicated. But you're never going to eradicate all the subclones in the population, because the tumor is a bomb. And by the time the tumor becomes, even a centimeter cubed, you're going to have a great deal of heterogeneity within the tumor, because the different subpopulations of cells within the tumor that's evolved in this kind of Darwinian selective way over years, they will all express different cohorts of mutations. And we know this. I mean, we know that depending on the tumor type, lung tumors, for example, can have more than 100,000 mutations. And those mutations are not evenly distributed within the tumor population. So you have subclones of cells within the tumor that have very particular mutations that are private to those subclones. And then there are mutations that are more common to all the cells in the tumor. So it's complicated. I think what happens a lot of the time is, and we know this is the case in most solid tumors, so the immune system's recruited in as a result of, say, the necrosis, gets activated. If there's strong tumor antigens, those T cells will eradicate those cells, but not all of the cells are going to share the same mutations. So now you will allow survival of the cells that don't have strong tumor antigens. So they will regrow the tumor. You will get then further wave of immune activation. And eventually, you're either going to eradicate the tumor completely, in which case you never have a tumor, you don't know about it. Or when a tumor does arise, by definition, it's already escaped your immune system, right? So there's no tumor antigen there that, a T cell is capable of effectively, seeing anymore, because all that immunoediting has happened over years. And now the tumor is just benefiting from any further incursion of the immune system into the tumor. Those immune cells are seeing the tumor as simply a wound. And what do they do to a wound? They heal it. So I hope that's not too simplified, but that's the way I see it. I don't think
Thank you.
there's a magic bullet in any of this. I think it's a kind of a very predictable biological process that happens in any tissue when there's a lot of tissue disturbance. Our immune system doesn't go in and make, again, a completely binary decision. It makes a decision based on what it encounters. And the problem with tumors that are selected over a long period of time is there's no major non-self or infectious entity there to basically aggressively attack, particularly when the tumor becomes really clinically detectable. So it does what you'd expect it to do with any wound, and that it tries to, neovascularize the wound and trigger, wound healing within the wound. So.
Thank you.
Agatha.
Hi, are you listening to me? Okay.
Yes.
Thank you so much for the lecture. It was really inspired. And my question, Professor, is actually kind of a curiosity, because I was wondering it is possible to transform or shift the necrotic process into an apoptotic process, and what could be the consequences for the body.
To shift which process exactly?
I think the beginning of the necrotic process actually turns into an apoptotic, and actually, if we could change this in the body and make this process not to be so hard to the body, like not getting into a necrotic, but apoptotic.
I think in most cases where you get necrosis, as I mentioned, it's usually these are usually extreme stimuli, and so I think it'll be quite difficult to dial those down to get an apoptotic outcome where you're getting a necrotic outcome. So yeah, I think it's I think a lot of research really that's been done maybe is more in the direction of switching on apoptosis when the cell fails to die. For example, in a cancer chemotherapy situation where you treat a cell with a drug, you hope to kill all the tumor cells, and instead what happens is all of the cells gets exposed, let's say, but only a fraction of them die by apoptosis. So the rest of them manage to resist that dose of drug, and they avoid entering apoptosis. So I think in that situation, yes, I think it's possible to lower the threshold of those cells to entering apoptosis so that more of them enter apoptosis. And as an example of that, there are drugs called BH3 mimetics, and those BH3 mimetics, they neutralize the pro-survival members of the BCL2 family, and they effectively lower the threshold for apoptosis so that then more cells of the tumor die. But in terms of reversing necrosis, remember the things I said that cause necrosis are things like burn injuries, very high concentrations of toxins. In order to dial those down, to getting apoptosis as a result of necrosis, I feel that would be quite difficult to achieve. But maybe it's possible, but I would be less optimistic than the reverse situation where cells are not dying and you want to encourage them to die by apoptosis. I think it's easier to neutralize the inhibitors of apoptosis to allow cells to die in a situation where that would be desirable than it would be to kind of reverse necrosis. It's kind of like saying, and okay, this is an extreme analogy, but if somebody, let's say you set off a bomb, how can we protect people in situations where like a bomb has gone off? It's going to be very difficult. Like necrosis is really a bomb being dropped on the cells. Of all the things I know that we use in the lab, and we've been working on cell death for over 30 years, the kind of things we have to put onto cells that cause necrosis are really extreme. The vast majority of cytotoxic drugs, if you literally took almost every compound off the laboratory shelf and start titrating it onto cells, the vast majority of compounds, as you titrate up the compound, they would induce apoptosis. You'd have to go very high on the dose to get necrosis. So I wouldn't be very optimistic about treating the cells with those doses and then getting apoptosis as an outcome instead. Be quite difficult to do.
Shay,
Okay, thank you.
Shay,
sorry.
Yep.
in regards to your current interests, maybe a very naive question, is there a point of no return to senescence? Thinking about this protection of the bone, for the bone?
Is there a point of no return
Yes.
beyond which cells don't enter senescence, or they go into apoptosis then, you're saying? Or is there a point of...
Yes, my point is, if a cell decides to not to die and to go to the senescence, is there a point of no return for these cells?
I think when they enter senescence, they are functionally dead in that they don't proliferate anymore. They're seriously wounded. I would look at senescence a little bit like, being very seriously wounded and on life support, right? So you're in a bed hooked up to, a life support machine. You're still alive, but you can't do much.
But they don't induce inflammation, at least.
Well, so yeah, so I mean, certainly you can manipulate potentially the
Yes.
senescent cells, and what we're looking at is what are the molecules that dictate the choice between getting into senescence or dying, and in fact, we're finding that many of the regulators of cell death, if you start, basically manipulating them, instead of dying, you can start getting into senescence instead. And so, many of the same regulators that regulate apoptosis also regulate senescence. And that's the picture that's emerging. I certainly think it's possible to prevent senescent cells secreting inflammatory factors by manipulating some of the molecules involved. And we're looking at that as well. But this switch is quite fascinating because senescent cells are really, it's only something we've started to look at in the last few years, but they're extraordinary cells. So we can take a dose of, for example, cisplatin, and treat the cells with cisplatin. And in previous years, we would always be just looking at how cisplatin induces apoptosis in the cells. And I'm talking also about you can use, daunorubicin or doxorubicin or many other DNA damaging agents, etoposide. They're all used to treat patients. So it's a very relevant question. So you take a dose of the drug, and 50% of the cells die. And in the past, we would have been examining the cells that die, and we would, follow them for 2 days, and then we would, do our various measurements and look at the molecules to regulate cell death. What we weren't doing is looking at the cells that didn't die and asking, what do they do now? And so now we're doing that. So what we do now is we treat the cells, we throw away the dead cells. Basically, cell death is maximal about 48 hours after the treatment with cisplatin or etoposide or daunorubicin and so on. And okay, now we wash away the dead cells and we look at what happens to the rest of the cells, and they all enter senescence, but they become enormous. They become about 6 to 8 times the size of a normal cell. They become incredibly enlarged. They start secreting a lot of inflammatory factors. They show a lot of other changes to the cell architecture. So they're beasts, these cells. And so we can tip the balance. For example, if we remove FLIP from those cells, C-FLIP, which is a regulator of death receptors, now they all enter apoptosis. None of them get into senescence. And
so interesting.
There are other molecules. So we know that, molecules like death receptors are very important in dictating this kind of decision to enter senescence or enter apoptosis. And so we're quite interested in, it's been suspected for quite some time now that One of the problems of chemotherapy is that the senescent cells that arise as a result of the therapy, they start to promote inflammation within the tumor, and that can make the tumor more aggressive, more metastatic. And so there's an approach now to try to eradicate those senescent cells by identifying the factors that senescent cells rely on, to survive. And certainly, some important cell death regulators are very important for senescent cells, sustained senescence.
So interesting.
Yeah, quite an interesting
Great. Gu, before
No,
Isabella.
yeah, no. I was going to say something about the previous question. So Seamus answered regarding the conversion of necrosis to apoptosis. Okay, but I would just mention pyroptosis, for instance, to apoptosis in terms of necrosis type of death. Okay, and the comment would be that not always, moving from a necrotic type of cell death to a apoptotic type of cell death would be beneficial to the organism. Okay, if the only form of cell death beneficial to the organism was apoptosis, then probably, during evolution we wouldn't get any other sort of cell death. Okay, so as Seamus said, it's all context depending. So depending on the infection, it's quite important that we have necroptosis or pyroptosis. To deal, mostly pyroptosis.
Yeah, and the thing is, you only want apoptosis where, as I said, you don't want involvement of the immune system, where involving the immune system would not be beneficial. So where cells are being turned over quite naturally as part of a just homeostatic process, where there's no infection, there's no major perturbation of the system, then apoptosis is appropriate. But if you have a viral infection or you have a burn injury or a very severe, tissue disturbance, then you want necrosis because you want to alert the immune system, and then you want to activate the immune system so it can ramp up immunity if there is an infection or there's a need for an aggressive wound healing response in that situation. So, of course, necrosis is very beneficial, and that's why we have DAMPs in order to activate the immune system during necrosis, and that's why we have pyroptosis. I don't believe in necroptosis in vivo, I have to say. Sorry, Peter. But at least there's very few situations where, there seems to be clear evidence for that. But, so look, there's a lot of new modalities of cell death that's been suggested in recent years. I'd be a little bit, ferroptosis and panoptosis and so on. I'd be a bit skeptical of those. But there's no doubt pyroptosis is very important in infectious scenarios. You want that to happen. You want the cell to burst open and release a bolus of IL-1 beta and IL-18 in an infectious setting because you need it there. You have bona fide evidence of infection where you get pyroptosis. But if cell death is happening during, the selection of the T cell repertoire in a sterile thymus during embryogenesis, I don't think you want inflammation in that situation.
I fully agree, Seamus. That's why I didn't want to talk about necroptosis.
Really?
Yeah, but he wanted to talk about ferroptosis.
Ferroptosis, yes.
Oh my God. There's a new form of cell that we're working on. It's called chocolaptosis, where you eat too much chocolate and then you get a lot of cell death. I have to say I'm a little bit worried about the definition of or the introduction of all of these new cell death terms. Evidence. Ferroptosis is a good example. There is no molecular regulation in this pathway. It just looks to me like a detoxification pathway. And if you get a lot of lipid peroxidation and you take away GPX4, the cell just bursts.
Of course, yeah.
I don't see how that's a cell death pathway. I think that's a detoxification pathway that when it fails, you just simply rupture cells.
Yeah. But also TNF is a detoxification pathway.
TNF.
Yeah.
Well, I think I'd like to hear your arguments for how that is, but I think TNF
So
is
here's the deal. Here's the deal, Shane. Tomorrow you come and listen to Peter, and then
No,
I think
argue.
I think there's scenarios where you get, look, I think there's a good argument that necroptosis is, you know, at least a cell death routine that can be invoked in certain situations where you perturb apoptosis and then you get this RIPK3 MKL, you know, poor. It at least looks like a cell death, an active cell death process, but ferroptosis, you know, there's no pore, there's no, you know, active regulators of the process. It's just if you take away detoxification routine, cells burst. I'm not sure how that's a cell death pathway, you know what I mean? You know, it's
but there are disease conditions where redox metabolism is affected, and there it occurs even without any stimulus.
yeah, I think yeah, true.
For
I
example,
mean,
ischemia conditions, there you don't need any stimulus, and you can protect with inhibitors of ferroptosis. But I'm not going to talk about ferroptosis.
I'm just saying I think that's simply necrosis.
No, but there are disease conditions.
Well,
There are
it's
conditions.
either a cell death or a cell survival.
No, but that's it.
I think it's a cell survival pathway that when you eliminate it, cells die by necrosis.
Yeah, but in
I think
most cells, for example,
is
most
problematic.
cells will not, for example, all the dead domain receptors, most cells, when stimulated with dead domain ligands, do not die. So there are continuously protective mechanisms also in that way, in that condition. So that means you need continuous survival signaling to cope with all those challenges that at a certain moment may. So you need continuous detoxification mechanisms.
But the difference there, Peter, is that when you take away the, let's say, the inhibitors of death receptor induced
I fully agree. That's a
then you get
sensitization.
activation of a series of enzymes that actually, you know, orchestrate the death. Whereas when you take away GPX4, what's the mechanism of death there then? It's not a series of enzymes. You just get massive lipid peroxidation in the cells. So I do think there's a difference. For example, you know, if you remove actin from the cell, you'll kill cells, right? So is that any mechanism to disturb actin a novel cell death modality?
No.
I mean, you know what I mean? Any important protein, if you remove it.
But we are talking not about actin, we talk about redox metabolism.
I know.
And redox metabolism can go, this is not a protein, this is about redox reactions. And that's very extremely important to keep your cells in a good shape.
No, but all I'm saying is that I'm not saying that's not important. It is important.
Yeah.
It, to me, it looks like a detoxification pathway.
Yeah, absolutely. But
cell death pathway, if you know what I mean. It's a detoxification pathway which, when it fails, cells die. I don't see the molecular effectors in the same way that I see with apoptosis or with necroptosis or pyroptosis. But
so, guys, when you hear Peter saying absolutely means no way, Jose.
I can see that. I can see the nodding. I know what the nodding means.
All right. So next question, guys.
But I think the majority of the questions are related to subject that we already discussed here. Don't know if there is another.
There was a question there about.
I think Maribel has a question.
Maribel.
There's quite a few in the chat.
Yes, Maribel.
Go, Maribel. Turn on your Mike and ask Seamus a question. So Seamus, this day, Maribel, Seamus speak a very good Spanish, so you can ask him in Spanish.
Habla un poco.
Good afternoon, profesor. Mi pregunta es si la presentación
No, don't
y
speak
la...
in Habla in English.
Okay.
No español.
Bon, thank for the presentation, Professor, who gives your opinion on the currently state of melanoma treatment in a manipulated cell death pathways in the clinic. Clinic.
Melanoma treatment.
Yes, the manipulate yes, necroptosis como apoptosis, pyroptosis.
I think it's a complicated, question. We know that melanomas, notoriously difficult to treat. RAF inhibitors, BRAF V600E inhibitors have obviously, given tremendous progress over the last decade or so, targeting the BRAF V600 mutation. I think the drug is called Zelboraf or vemurafenib, and that certainly helped. But in terms of the modality of how melanoma cells die in a treatment setting, whether it's apoptosis or necroptosis, or I don't have an opinion on that. We haven't worked extensively on melanoma, but these cells are metastatic, invasive melanoma cells are known to be notoriously refractive to most chemotherapeutic drugs. So when they do die, when chemotherapy is effective, I'm not sure what the modality of cell death is there, because really that's not a literature I've been following. So I don't know.
Thank you.
All right, guys, I think it's already over 4 p.m. and 4 p.m. here. For Seamus is around 8. Is that right?
Yep, it's 8:05, yes.
Okay, so just would like to thank you very, very much again, buddy. So Seamus has been in all editions of our course since the beginning. And tomorrow I invite you at 8 a.m. to know a little bit more about necroptosis. Although I have a feeling that Peter will also talk about ferroptosis. Just slip in some other. And I invite Seamus to participate as well in this debate, fierce debate. All right.
Thanks, Shay.
Thank you very much again.
Again.
And
So,
yeah, just wait a bit here, you and Peter.
Yes.
yeah.
So I want to hear
Bye, guys.
about the game. Yes, exactly. Bye, guys. See you tomorrow.